Alright, super nerds. So today we're going to talk about DNA. We're not going to go into so much of it as a form of evidence, except for a brief summary when we talk about the Green River Killer. But we will also talk about mostly the structure and the composition of DNA. So what is it made of and how is it put together? So by the end of this, you should be able to explain the details of the deoxyribonucleic acid, and you would also be able to outline how a change in technology allowed the conviction of the Green River Killer, Gary Ridgway. Alright, so deoxyribonucleic acid, which we are now going to call DNA, what is it made of? It is a macromolecule, so it's a polymer that can be anywhere from 10 to 100 million monomers long, and the monomer is called a nucleotide, and this is our nucleotide here, and you'll see that the nucleotide has three structural components. So they have a sugar, which is a deoxy, so it means it's had an oxygen taken away. A deoxyribose, which is a pentose sugar, so a 5-carbon, which is attached to a phosphate group and a nitrogenous base. Now the C1 attaches to the base up here, and the C5 attaches to the phosphate group. Now the phosphate group itself is phosphoric acid, which has had the hydrogens removed, and each phosphate group bonds with two deoxyribonucleic acids, or deoxyribose molecules. There are four nitrogenous bases or nitrogen bases, and they are sorted into two pairs. You have the purine bases, which are adenine and guanine, and the pyrimidine bases, which are thymine and cytosine, and their structure looks like this. So these are your purine bases, and these are your pyrimidine bases. And you can see one here is a six-pointed ring, so a hexagonal ring, and this is a hexagonal ring with a pentagonal ring attached to it. And that's the difference. So what about structure? Well, basically, it's a three-dimensional structure, and it has a double alpha helix with deoxyribose phosphate chains. So that's so deoxyribose phosphate. That's what makes up the chain. That makes up the backbone, and the bases point in towards each other and pair up in the center, and that's what gives it the double helix. So we have one helix going around, and we have another helix going around as well. The two helixes or helices are held together through hydrogen bonding on the base pairs. Now, once again, we've got that case where the hydrogen bonding seems to be one of the main types of bonding we do in chemistry at our level. The adenine and thymine, there'll be a picture of this in a second, are held together with two hydrogen bonds, and the guanine and cytosine are held together with three hydrogen bonds, and that looks like this. Now, this is a structure that you need to know and understand and be able to reproduce. Now, essentially, it looks like this. So it goes phosphate, sugar, phosphate, sugar, phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar, and you can see that the deoxyribose are pointing in opposite directions. This is one of the reasons they're called complementary strands, like they sit opposite each other. And we also have the complementary base pairs. Here are the cytosine and guanine with the three bonds, and here's the adenine and thymine with two bonds. And here's a little summary. Remember, you need to know the structure of these shapes as well, but this is a little summary. All right, so let's have a look at the Green River Killer, or Gary Ridgway. He murdered 49 of his victims between the years of 1982 and 1988, and he confessed to twice as many. He would strangle women, prostitutes usually, or young runaway teenagers, often by hands and occasionally with ligatures. But this is not the physical evidence which became key to his conviction, or the deal he had to prevent the death penalty. The first 5 were found near Green River, and so that's this area here, and the press gave him his nickname. He had an inappropriate sexual relationship with his mother, and he was also physically and emotionally abused within the church as a child. So he was a victim of abuse from a very young age, and he also had a very low IQ, which is actually quite rare amongst our serial killers. And he was always described in school as being quiet in nature. Now, he became a suspect in 1983, or 82 actually, and was arrested under caution due to his truck matching the description of the scene when some of these women went missing, and his location at the time, both where he worked and where he lived. He was not charged at the time, though, due to insufficient evidence. Now, the murder would return to his victims who had sexual intercourse with the bodies. Normally, we would have left this detail out of class, except he left semen deposits. Now, this is essentially what was his undoing. He was arrested in 1987 by the FBI, and so with many suspects, and hair and saliva samples were taken, and these were used to match blood type to the semen, which narrowed it down to a potential 100 or so killers. Now, in 2001, however, the technology had changed, and we now had an improved way to amplify DNA called PCR. And we'll go more through this in our next lesson. So in 2001, the evidence was reviewed and reanalyzed, and Ridgeway was the only match from hundreds of suspects arrested in the mid-80s. Because he was the only match, this meant he had been the one who was the killer. He was the one who the physical evidence linked. Now, not only was he the one the physical evidence linked to, they had him quite convincingly for 4 to 5 murders, and being a death penalty state, he offered a deal where he would confess to 49, and 49 with evidence. And because he had the evidence for 49 of the murders, they were able to convict him with those 49 murders. He confessed that there was at least another 50 more, but they couldn't find the bodies. So he now is serving 49 consecutive life sentences in jail. And that's how the DNA worked. Now, it's important to remember, one, that the key evidence was DNA evidence. It was useful because of a change in technology. And that's a dot point in several issues here, so we need to understand it was useful because of a key change in technology, advancement in technology. All right, well, we'll see you in class.